So I'm very happy to introduce the next speaker, Dr. Nancy Yacovzada. She's a senior medical data scientist in the Weizmann Institute of Science, where she also did her PhD together with Eran Ornstein and Eran Segal. Nancy is actually leading the AI research in the lab of Eran Ornstein, and she will talk about organellomics, AI-driven deep organellar phenotyping of the human neurons.
Can you hear me? Right, so I will talk about how we combine molecular biology and computer vision at Weizmann. It's not working. So the common ground of the people in the picture is that they were diagnosed with brain disease, and more specifically neurodegeneration, where neurons degenerate and die. Can you please? And one of the challenges in studying brain diseases is the fact that in opposed to cancer, where you can do a biopsy of the liver or the pancreas and just get the cells that you want to study, in brain diseases, you cannot enter the living brain of a patient. And therefore, for decades, studies were made on post-mortem tissues, where you, once the patient died, dissect different tissues of the brain. But post-mortem is actually a snapshot of the disease at the end of the disease, and it limits our ability to study disease development. And we are an integrated team of computational scientists and experimental biologists. So the biology side is led by Dr. Lena Molitor, and the team of the data scientists, which are here in the crowd, Sagi Crispin and Noam Rutberg, led by me, working with Professor Eran Ornstein at Weizmann. So how do we study brain diseases? So one of the big advancements in biology was iPSCs, which the lay term is stem cells engineering, where you can take a simple biopsy from the skin, and then what you do, you reprogram the cell to go back to be a stem cell, which is the origin of all the cells in our body. And then you can differentiate them, which in our language means to program them and engineer them to become neurons, and then you can grow them in a dish. And once the neurons are mature, what the experimentalists can do is basically abuse them and try to mimic a disease. So for example, you can go to genes that are known to be associated to the disease and insert using another great advancement in biology, which is CRISPR insert mutations. You can do drug screen and look for new treatments for the disease. By the way, most of the diseases that we study don't have a cure. So then you can do high throughput and screen different treatment for the disease and see how the survival of the neurons change. How can you prevent them from degenerating and dying? Because neurons are basically the only cells that do not regenerate in the body. What are organelles? So every cell has different organelles, and each organelle has its own function, and some of the organelles are known to interconnect and do important stuff in the cell together. So we know that organelles also play a key driver in cell survivors. So if, for example, the cell stops to generate energy, then the cells start to die and the neurons degenerate. So what is the data that we work on? So we're interested in how organelles change, let's say when you have a genetic mutation or if you take a drug. And we are able to do high throughput, not me, the biologist, to generate high throughput data, so around 3.5 million images. Currently it's actually double the amount of data that we have. And you can do experimental conditions. So we did 9 different experimental conditions in human iPSC-derived neurons. So the unmet need in the field when we started working on the project was first to work on images of neurons, because neurons are not simple round cells. They're connected. They have their extensions and neurons between each other. And the biologists wanted to be able to get features of the images. And when we say organelle localization, so in opposed to what you think that localization means the position of the organelle in the cell, so no, it means everything that the pixels actually encode, which is the intensity, crispiness, shape, size, number of objects. So we want to be able to encode an image. And we also want to be able to give the model two different experimental conditions. And once we have the features, we needed to develop tools to visualize those latent features and generate plots that biologists can understand. Another unmet need was that currently the field understands that, as I said, there is a connection between the different organelles. Some of them play together and do important stuff. So how can we study all the organelles together? Because current microscopies are very expensive and they are limited to only color 4 channels in a time. So you can look only on 4 organelles in an overlapping cell. So how can we computationally overcome this limitation? And I will talk about it briefly soon. So our architecture is self-supervised VQ-VAE. So as you can see, we have encoder and decoder, but some other modalities in the model that I will discuss. And the model outputs 2 different representations. Those are the colorful cubes that you see, and each of them is used for different purposes of analysis. So let's start about what's the VQ, vector quantization. So a regular encoder gets an image, learns how to represent the image in the latent space, and then the decoder learns how to reconstruct the image. But with the vector quantization layers that are added to the architecture, what you basically do is you map the continuous latent space that you have, the continuous vectors, into discrete codes. We term these codes code words, and they are actually a part of something that is called codebook. And the idea is that in inference, once you have an image and you encode it with the encoder, you then transform it to a discrete representation, which is more compact and understandable. So why understandable? Because then we can go and look, for example, in the picture of the dog here, we see that the yellow code words encode for eyes, and the blue ones encode for the nose. So you do augmentations, and you train the model, and you learn the code book during the training. And you want to be able to decode any kind of nose, any kind of eyes. Another element in our architecture is that we use 2-level latent space. So we have 2 encoders, 2 decoders, and 2 code books. This is work done by DeepMind, presented in NIPS in 2019, which basically gives you the 2 cubes that I presented. One is the local representation of the image, and one is the global. And the idea is that each of them encode different information of the image. So for example, if you train the network with different cell types, so the global information is, first of all, which cell type is it, because neurons look very different than microglia cells, for example. But the local information can talk about the geometry of the localization of the specific organelle. So we said that the architecture is self-supervision. So how? First of all, the experimentalist that generated the data can tell us which conditions and which organelles he stained. This is the label that we have about the data, but it's a soft label. We don't know whether the mutation actually changed something in the representation of the organelle in the images. So the first task, as I mentioned, is to encode the image for neurons, and you measure this by a loss of the MSE between the original image and the reconstructed image. But this is what really drives the algorithm to work on the task that we want. So we have a pretext task, which use the label and the encoded representation, the latent codes that are discrete, and tries to predict the label giving the representation that we have from the tool. So then this is measured by cross entropy because this is a multi-class problem. And those two terms are introduced to the general loss function of the architecture, and this is what actually encouraged the model to learn the localization-specific patterns, meaning to eliminate things like noise that you have between different experiments or the background that you have in the images. So I will give you a few bites of what exactly we're doing with the outputs that you get from the model. So we did the first map of all the organelles in neurons, and this basically can generate new hypotheses about two different organelles that we didn't know that have any connection and that they actually operate together. Another thing is I told you that we have the VQ in the architecture, which can give us a great gift for explainability, because we can say for every given organellar condition which codebook vectors, which code words were used to decode an image. And then you can go and generate a histogram of the usage of the different code words from the codebook, and basically to turn the histograms to densities. And those densities can be compared between different experimental conditions, between different organelles, and this generates lots of hypotheses for the biologist. And Noam developed a retrieval algorithm that basically can go back and say for a specific group of code words, what is a representative a represented image that was using this specific code words, and then we give it back to the biologist, and they go and do additional experiments. Another approach that I mentioned that we wanted to tackle is to overcome the limitation of the microscope. So Sagi Crispin have developed an approach which is being approved as we speak, but this is the one that we have in the bioarchive. That basically use in silico approach that concatenates images of different organelles and do matching, and then you have basically different encodings of the different organelles. You concatenate the vectors into one single point that represents a condition, and then you can go and do a UMAP. So one of the results we have is about the ALS genetics. So in ALS disease, there are different mutations that can lead to the disease. And in blue, you can see the healthy control. This is neurons without any mutation. And we can see that some of the mutations, the purple and the green, don't really show an effect. But when you look at all the organelles together, we see the brown FUS, it's a non-ALS gene. We see a major effect of all the organelles. So here you basically supercompose information that you cannot do only in experimental work. I want to again thank Noam and Sagi, which are here in the crowd, and you can feel free to reach out and ask questions. And I want to thank you for listening.
Thank you very much, Nancy. Questions from the audience? You always have a question, so.
Is there any plans to try to generalize this to also include video and not just words, and then potentially multimodality, including, say, sensors or robot arms to play around with it and really try to dig deep? Or is that like we're not there yet?
I wish. The limitation.
Just please repeat the question for the recording.
So the question was, can we extend the vector quantization approach from images to videos? And you're right, in videos we see way more information, but the current limitation is to use iPSCs of neurons and the CRISPR genetic mutations to do live imaging. So we, the computational side, are eager to start working on videos. Currently there's challenges in the experimental side. Wow. So explainability is a major problem. One of the things is that we say that an organelle changes or all organelle change upon some experimental condition, but we cannot provide the biologist an explanation to what changed. So then we go back and see that you actually see a signal that was inside the nucleus and upon some condition goes outside. And we know that this caused the neurons to die. But we still not there in terms of computational to the tool to give those. And I think that this is actually not in Weizmann we have a lot of resources of computation. This is not the problem, the data. It's very expensive to generate the live video that he mentioned. So we don't have exactly transitions, so we need to do different time points. And you see we are 3 data scientists with 4 experimental biologists. You need way more people to generate the data. So
I got a cue that we have to move on. So
okay.
Thank you very much
Thank
again.
you.